So hello friends and my dear students, today we are going to study the part DNA replication or prokaryotic DNA replications. This is the new unit. Before that already we have studied the DNA as a genetic materials and RNA as a genetic materials with the help of different evidences. And apart from that, we already studied the properties of DNA, chemical stability of DNA, and prokaryotic chromosomes, E. coli as a genetic models. It means that much part is already completed. So today we will start the prokaryotic DNA replications. So first part, that is the introductory part of the prokaryotic DNA replications. So DNA, it is a genetic material which transferred from parent to offspring or which transferred from one generation to another generation. It carries genetic information from generation to generation. It carries genetic information, whatever the information regarding the genetical means what DNA of the parents and offspring, they are the same. That's why whatever the information regarding that, it transferred. Second thing, the responsibility to preserve the identity of species over millions of years. Yes, no doubt, because the DNA of parent is same as the DNA of offspring. Like that, the DNA of father, grandfather, or it is the DNA which long from millions of years, it preserve its identity of the species. Means with the help of the DNA we can identify the peoples, they are belongs to such family. It means there is no side effect, there are no any changes in the DNA. That's why its own identity is in the DNA last from many years. DNA may be regarded as a reserve bank of genetic information or memory bank because the DNA, it transfers, it is a hereditary characters which transferred from one generation to another generations, and whatever the information regarding the characters, whatever the information regarding the genetic, or whatever the informations regarding that species, it remains present in the DNA. That's why they are called as reserve bank of genetic information, or we can say that memory bank, just like memory card. Some viruses contain RNA as a genetic material, but DNA is more stable than RNA. As you know, with the help of a number of experiments, we proved that DNA is more stable than the RNA. RNA is not that much stable. After a period of times, RNA is degraded, or there are so many side effects on RNA. DNA is more suitable molecules for long-term repository of genetic information. It is very suitable for handling. Handling means for transformations, and is a long-term repository of genetic. Long-term means from millions of years it transfers its character from parent to offsprings without any change. That's why they are called as long-term repository of genetic information. Whatever the genetic information, they remains present in the offspring, they remains present in the parents. Like that, millions of years, whatever the genetic information, it constantly without any changing, it transferred from. That's why they are called as long-term repository. So in the prokaryotic DNA replications, replication means what? It is very important part. Replication means in general we can say that from old DNA there is the synthesis of new DNA. From old DNA there is the synthesis of new DNA, and this type of replication generally found during the cell divisions, because prokaryotic or bacteria, they generally divide binary fission manner, means one cell divided into two cells, and within that two cells, two daughter DNA or two daughter new DNA cells, they are produced from old DNA. And central dogma, that's why they are called as central dogma of life. Replication is called as central dogma. And central dogma, it is also one of the indications, or it is one of the experimentally process that is in the replications. The biological information flows from DNA to RNA and from there to proteins. Means how this information flows, because whatever the genetic information which transfer from generation to generations of parent to offspring, no doubt, but how it flows from DNA to RNA or RNA to proteins. And this is one of the process, or these are the reactions through which DNA is converted to RNA by a transcription process, and RNA is converted to protein by translation process. These processes are not that much easy, just like we are observing on the screen, because there are number of involvement of enzyme system, number of proteins, or number of other parts of the cells, they are involved during the synthesis of DNA to RNA and RNA to proteins. And this type of process that we can say central dogma of life. So DNA is a cell must be duplicated, maintained and passed down accurately to the daughter cells. It means that DNA is duplicated means one DNA is converted to 2 DNA during the replication process, and during the passing of such new DNA into the new cells, whatever the accuracy of that genetic information of the DNA, it remains the same, just like parent, it remains transferred into the daughter cells without any change. So when the cells divides, the daughter cells receive an identical copy of genetic information from the parent cells. It means that the replication occurred during the cell division. Just I given some idea or information regarding that. So I already said whatever the replications, it happens during the cell divisions. It means that one cell divided into 2 cells. During that times, what happens? The daughter cells, they receive the genetic information from the parent cells. So this is the another definition, scientific definition of the replication. Replication is a process in which DNA copies itself to produce identical daughter molecules of DNA with high fidelity. It means one DNA produces 2 DNA. One DNA produces its copy, just like a Xerox. Same, whatever the nitrogen base pairing in the old DNA, like that same nitrogen base pairing observed in the new DNA without any change in the sequences of nitrogen base pairing. That's why they itself produces identical daughter molecules of DNA with high fidelity. High fidelity means high accuracy without any change. So replications is a semi-conservative, because before the discovery of this type of replications, a number of scientists, they said the replications may occur by 3 ways. One is the conservative type, second one is the dispersive type of replication, and third one is the semi-conservative. And after the lots experiment discovery, finally replication is occurred by semi-conservative type, and dispersive and conservative type of replication cannot occur. That's why this is the final conclusions where the replication occur in semi-conservative. In the semi-conservative replication, what happens? Because DNA is made up of 2 strands. According to Watson and Crick model, DNA is a double-stranded helical structure. These are the 2 strands of DNA. We can also say that these are the parental DNA. These parental DNA, they are separated during the semi-conservative replication. You just see, These are the two strands. They are binding with each other by a specific bonds. During replication, the bonds are breakdowns, or these two strands are separated. On each strand, these are the two strands are separated. On old strand, there is the synthesis of new strand, and on this old strand there is the synthesis of new strand. It means that on these two old strand there is the synthesis of another new strand, and the old one DNA, it becomes 2 DNA into 2 daughter cells, like that. So on old strand DNA, whatever the nitrogen bases are present, just like whatever the synthesis of new strand, so the base pairs always equal adenine always pairs with thymine, and guanine always pairs with cytosines, and that type of pairing on a new strand or in between the old and new strands. It means that the exact same copies of DNA in the daughter cells like the parents, and new DNA, a new strand, or that is also we can say complementary strand. Both the strands undergo simultaneous replication to produce 2 daughter molecules. So when the 2 DNA are separated from parents, during that time at a exact time, at one time, what happens? On 2 strands there is the synthesis of new strands. It means that both strands undergo simultaneous replication and produce 2 daughter cells. Besides that, the DNA sequences by DNA repair, all organisms must duplicate their DNA accurately before every cell division. Means there is the accuracy, no doubt. DNA replication occur at polymerization rates of about 500 nucleotides per second in bacteria and about 50 nucleotides per second in mammals. It means that during the DNA replication, what happens? Because DNA is also called as polynucleotide. Polynucleotide means there is the chain of nucleotides. One nucleotide is made up of what? Deoxyribose sugar, phosphate group, and one nitrogen base. And like that, there is the pairing in the nucleotides. And there is the pairing of nucleotides. It means that there is the synthesis of DNA. And as per the polymerization rate in case of bacteria, what happens according to the research, say that what 500 nucleotides are synthesized per second in bacteria, and 50 nucleotides they are synthesized in the mammals. It means in the mammals the rate is low than that of the bacteria. Clearly, the proteins that catalyze this process must be both accurate and fast. It means that during the replication process, what? The proteinic nature of enzymes, they plays an important role. They catalyze what? The process accurately or fast. It means that a number of enzymes, they are involved in the replications, and due to the involvement of these enzymes, what happens? The reactions are catalyzes. You know, catalyst means it is one of the compound which are involved in the reactions without participation. Reaction is whatever the reaction is forwarded, reactions are carried out, but they are not involved. There is no their participation. They process carried out, but without any involvement. Speed and accuracy are achieved by means of multi-enzyme complex that guides the process and constitute an elaborate replication machines. It means that not only one type of enzymes involved in the replications, number of enzymes that we will see in the next lecture, where we are observing how these enzymes involved into the each reactions and how these enzymes proceed or catalyze the reactions and how the replications completed. Means that we are observing in the next lecture, where all these enzymes they are involved, but they are the multi enzymes and they generally elaborate what? Replication. That's why they are called as replication machines. Replications occurs in 5 prime to 3 prime direction only, because DNA is made up of, according to Watson and Crick model, DNA is a double stranded helical structure. It's the one strand of DNA, it starts from 5 prime to 3 prime directions, and another strand of DNA, it is a completely opposite. It means that the direction is the same, but another strand is the opposite direction. So what is mean by 5 prime and 3 prime? That is very important part, because most of the students they unknowns about what 5 prime and 3 primes. Apart from studying last from 12 to 10 class, 5 and 3, they are nothing but the numbers of carbon numbers of deoxyribose sugar. It is the number of number of deoxyribose carbon sugar, because it starts the carbon number 5 of the deoxyribose and it ends 3 carbon number of deoxyribose. Means start and end means the strand, whatever the start of the strand which from 5 prime, and whatever the end of that strand which is the 3 carbon number of deoxyribose. Replication is a simultaneous on both strand. Replication is a bidirectional, just I given. So on both strand there is the replications according to the Obey's base pair rule. The scientist always said this is the replication, and it is a bidirectional. Replication results in 2 daughter DNA strands. Each daughter DNA strands has 1 parent strands and 1 new strand, that we can say complementary strand synthesized. Hence this replication is a semi-conservative held by phosphodiester bonds, or semi-conservative replication. So friend and my dear students, this is the introductory part of the prokaryotic DNA replications. Or for replication, what type of the replication generally observed, and for that replication what type of the requirements. This is the general idea. Okay, in the next lecture we will see why semi-conservative DNA replication is accepted compared to another type of replications. That we'll see deeply or neatly in the next lectures. So friend and dear student, you watch such videos, they are very important. We are giving valuable informations in each videos. If you are studying well, if you are observing such videos, no doubt 100% sure says if you are watching, you will get 100% knowledge regarding this type of topic. So if you have any confusion, if you have any problems, if you have any questions about this, so I request you have to mention your comments in the YouTube channel, and we are here to give the answer of your questions. Till then, bye.